I'm Aryeh Warmflash. I'm an associate professor in biosciences at Rice University, and I am a co-organizer of the meeting Stem Cell Models for Embryology, together with Magdalena Zernicka-Goetz, which will be occurring in February in Pacific Grove, California.
So to my knowledge, it's the first meeting that it's exclusively devoted to bringing together community that try to model development of the embryo with stem cells. The mission of this meeting is to talk about evolution of different models and to look at them from many different perspectives and bring together everyone in that field, that new field, to be able to work and enthusiast each other about their models and their way of thinking so that we can really make a progress in that field for the next years to come.
This meeting is really for anyone who's interested in understanding early human development or who's interested in how we use stem cells to model early development. So that includes researchers in these fields, researchers in baby developmental biology who work with model organisms and are interested in how that might interface with work in human systems, people working with stem cell models of human systems and coming together to really understand the different systems and the advantages and disadvantages of them, as well as clinicians who might be interested in these systems for modeling things like infertility or birth defects, and then as well as people in industry who might be interested in developing treatments or diagnostics or therapeutics for things like birth defects or IVF, infertility issues. The field of modeling embryos with stem cells is a relatively new field. So over the last 10 years, we've seen a number of models developed, and those have been increasing in their sophistication. And also what we've seen over the last 10 years is people really starting to use these models to dig in and understand aspects of embryology and of human embryology that really wouldn't be possible to understand any other way. And so we're using these systems together with things like CRISPR gene editing to create recorders, together with single-cell RNA sequencing to generate these really rich data sets that can probe aspects of development that really haven't been probed yet, even in sort of more accessible model systems. And so it really is a time where the field is growing, is making incredible progress, and it's a great time for us to come together as a field and share that progress, as well as establish a vision for building on that in the future.
I myself am focusing on implantation stages of development, but we will be representing many different aspects of development at this meeting. And we would be going through models that were only recently created, maybe only published a few months ago.
And so recently, myself and other people in the field have developed models in which we use stem cells to create objects that either look like embryos or look like parts of human embryos. And we can use these systems to study aspects of embryonic development that are specific to humans. And also, because these systems develop in a culture dish, we can use them in ways that we really couldn't work with any embryos or any model systems because of the flexibility they offer in terms of perturbing them, in terms of growing the cells in different ways, and in terms of especially imaging the cells. So they're very accessible for imaging as opposed to mammalian embryos, which develop in utero. And so this is a really exciting time in this field, and it's a great time to bring people together to think about the commonalities between these systems, what we're learning from these systems, and also to think about, especially for human, ethical challenges for these systems and frameworks to work with them going forward. Discussions around ethical issues will be a part of this conference. We have speaker who will specifically be addressing these issues, as well as a panel discussion with that bioethicist and people working to develop these models to sort of get the ball rolling on that discussion and bring people from different perspectives to start thinking about a framework for working with these systems.
And now is really a time when the meeting is really bringing together so many different labs, so many different perspectives that would be together discussed, presented, and hopefully co-evolving in an co-evolving in an inspirational way over the next years. This is really within the tradition of Keystone Symposia that are extremely interactive, and they try to do the best to bring together speakers that might have not yet met. And that's very, very important when the new field is created, as it is with the stem cell modeling of embryology. So I am really so much looking forward to meeting many new people that are entering this field, and also introducing people to each other. And that's the idea, that this particular setting in California are excellent for that.